One of the ways we can compare closely related strains or isolates is with the length of a genomic locus called a VNTR, or variable number of tandem repeats. VNTRs are small regions in the genome where, usually because of recombination errors or stuttering in the replication machinery, consecutive repetition of a set of nucleotides occurs. This means that there's functionally a printing error that we can exploit when trying to measure relatedness between isolates. VNTRs are useful because they're at a fixed location in the genome, meaning that we can amplify them using PCR primers that are conserved across all of our strains. Although the location is fixed, because of the VNTR, our PCR product will vary in size, in units of 10 or 20 or 36, whatever the size of the repeated sequence actually is. We can then use that size to try and draw conclusions about how related our various strains are. We can differentiate amplified product size using gel electrophoresis. Before we can visualize that, however, we need to run the PCR. First, we're going to check the purity and concentration of our isolated gDNA, because this is going to factor into our later reaction calculations. We use a nanodrop, which is a highly specialized spectrophotometer that measures the absorbance of our gDNA solution at 260 and 280 nanometer wavelengths. DNA absorbs strongly at 260, while because of the presence of aromatic rings, protein absorbs at a peak at 280. This means that we're looking for a fairly high ratio, usually between 1.8 and 2, between these two numbers before we can call it pure. When we're assessing the quality of large pieces of genomic DNA, it can also be useful to run our extracts through a low concentration of agarose. If there's a little bit of smearing, that means there might have been some chromosomal degradation, which might affect our results further down the track. Now that we've ensured our DNA is pure and we have a decent yield, we can set up our PCR. Because we're looking at the same locus in multiple different outbreak strains, we like to set up something called a master mix, where we combine the constant PCR reagents in bulk before splitting them up and adding our variable genomic DNA extracts. We'll need a separate master mix for each VNTR we look at, which means one specific pair of primers per mix. We'll also need enough master mix so that we can run a primer-only, no-template DNA control. This will allow us to see if our reaction is contaminated or if our primers are annealing to each other. We'll go over the calculations we need to do this together in class. Once we've figured out the correct volumes of each reagent we need to add, we can start mixing our master mix. First, we add UV irradiated and filtered water. We'll need to calculate the correct volume to add because that will vary based on how concentrated our gDNA extract is. We need the quantity of DNA we add to our reaction to be constant between strains for our experiment to be valid, and we change the volume of water we add to compensate for this. Then we add some glycerol, which acts as a thickening agent and helps to lower strand melting temperatures because excessively high temperatures can damage our gDNA. We need to add plenty of dNTPs, which are the ink that the Taq polymerase needs to print new copies of our region of interest. Then we add our primers, an equal quantity of both our forward and reverse primers, which will anneal and amplify from either side of our variable region in our VNTR. Then we add our PCR buffer, which contains pH buffers and dicationic magnesium, which DNA polymerases need to function. Finally, we add our Taq polymerase, which does all the work for us. Before we thermocycle our mix, we need to add our genomic DNA. Because that is reaction specific, I like to add the correct amount in a droplet in our final reaction tubes, leaving one empty at the end for our no template control reaction. We need to make sure our gDNA extracts are at a constant volume because we've accounted for that volume in designing our master mix. This might require some dilution. Once we've done that, we can portion out our master mix, flick to mix, and give it a quick spin to remove any bubbles. We need to design the correct thermocycling conditions for our fragments of interest. For our purposes, we're going to go with a primary melting phase of 94 degrees Celsius for 5 minutes, which will allow our long pieces of chromosomal DNA to denature and split apart. Then we're going to cycle through the denaturation, primer annealing, and Taq extension phases as shown here. We wrap up our thermocycling with a final extension phase, where the polymerase has a chance to fix up any loose ends. Once our amplification is complete, we need to visualize our VNTRs using agarose gel electrophoresis. First, we set up our gels. Our amplicons are going to be fairly small, so we'll be using a moderately high agarose concentration, 1.5% weight per volume. which we've pre-stained by adding gel green to the agarose. Gel green is a fluorophore that will fluoresce when bound to double-stranded DNA, allowing us to see our DNA fragments in the gel. We set up the gel in the tank using end-formers and well-formers and gently pour agarose mix into the tray. Once that's set, we can insert it into our electrophoresis tank and fill it with our running buffer, TBE. TBE contains small ions that will facilitate the flow of current and DNA through our gel. Once our gel's in place, we can prepare our VNTR samples for electrophoresis. First, we add our size standard, which more often than not comes pre-mixed with a loading buffer. These DNA ladders are controls that help us visualize the size of our amplicons. On a piece of parafilm, we drop a small amount of blue juice loading buffer, to which we add some of our reaction of interest. We gently pipette up and down to mix, and then add it to our wells. This blue juice contains glycerol, which is viscous and helps the DNA sink to the bottom of the wells, and bromophenol blue, which is a negatively charged blue dye that will allow us to visualize the movement of our reactions through the gel. Once our gel has been set up, we can connect the tank to a power pack and run it. Ensure that it's set up so that our negatively charged DNA is running to the positively charged anode. A handy mnemonic is run to red, back to black. You can check that it is set up correctly, as you will see fine bubbles evolving at the cathode. If you have set it up incorrectly, you will see the blue dye front move backwards, and you should adjust immediately. Now that our gel is run, we can take it down to the UV illuminator and visualize it.